I'm going to introduce you, Helge Rask-Anderson, a dear friend of ours. He spent years with us in 1988, 1989. It was our pleasure to have him at the Institute doing much of his early work. And I know Daryl has visited him, and I just learned that they did the first auditory brainstem implant in 1993. It was the first one done in Europe when Hitzelberger and Brackman went over and performed the surgery. So I can't tell you how thrilled I am to have you on with us, Helge. As I said, you're a dear friend and a colleague and a pioneer in the area of inner ear research. So Helge, we're anxious to hear what you have to say.
Oh, thank you thank you very much, John. It's a real pleasure and honor that you invite me to speak a bit about our research in Uppsala in Sweden. First, as I said before, I want to thank you because of the possibility to stay with you in 1988 at the house clinic, and Daryl and Bill, who came to Uppsala and to do the first ABI in Europe in 1993. That was really spectacular. So thank you from all of us in Uppsala in Sweden. And you know, Uppsala is located in Sweden. It's not a country close to Austria. I think some believed that when I was in LA. I don't think it was President Reagan. But as you all know, it's up in the north, up in the cold north. And Uppsala is located a bit north of Stockholm. And the university hospital had the first professor as Robert Barney. You know, I'm sure you know him. He was our first ENT professor, and he got the Nobel Prize 1914, but he was in Russian prison that time, so he came back 1915 and received the prize. So he was the first professor discovery of the caloric response. So I thought I would tell a little bit about our human research we do in Uppsala about super resolution microscopy and scanning electron microscopy, synchrotron studies we do together with Canada, and also now we analyze the gene expression in human temporal bone sections. So in Uppsala we have a large collection of corrosion casts. That was how I started 50 years ago, and we have 440 specimens where we can look at the anatomical variations, and they are also digitized. When I came back from Los Angeles, I joined the skull-based group, and we did some petroclival meningioma and with a transcochlear route. And actually, I saw my boss drilling through the cochlea after facial nerve rerouting. And I asked, why do you smash the cochlea? Why don't you take it out? And said, yeah, we can do that. So we got ethical permission, and we actually were able to get the whole cochlea out, which is a pretty normal cochlea, which is may look a little bit strange, you know, but that made it possible for the first time to do a real nice scanning electron microscopy study of the human cochlea, which you can regard it as a normal cochlea because the tumor infiltrated sometimes the nerve, but not the cochlea per se. So if you look with higher magnification, we can see the beautiful human organ of corti with a tectorial membrane and the Reisner's membrane here. Higher magnification, we see this these outer and inner hair cell, the efferent supply, nerve supply, and even higher magnification, you see the beautiful cigar-like outer hair cell with a prestin molecule sitting on the membrane. the extensive efferent nerve supply through the tunnel of Corti. You see here, you see the inner and outer pillar cells, and you see this beautiful anatomy. And the way we did it was that I took this actually to Innsbruck, and because they were fantastic to put a very thin layer of palladium gold on it to get high resolution. So I think that was very, very fortunate. We look at the tectorial membrane and see how beautiful it is shaped and different over the frequencies. We see the hair cell rows, which are quite differently organized compared to animals. You know, they are not as straightforward organized in humans, which is important. And this is not an artifact. And we can see how the stereocilia have different lengths depending on frequency. At the high frequency, they are shorter, and they are longer in the low frequency region. At the same time, we see this beautiful tectorial membrane, the structure that varies all along the cochlea. With a low frequency, you have these small processes that faces the outer row of the hair cells, stereocilia, and this is the more robust high frequency tectorial membrane. This is a bit surprising that you have such a variable shape, and this is 100,000 time magnification of a human inner hair cell. So this is where the human hearing sits, and you can see they look like you know, corns, you know, with this proteoglycans and glycoproteins sitting on the surface of the membrane of the inner hair cell. So and then we come to the spinal ganglion, which is absolutely fascinating, that the human spinal ganglion is entirely different from other mammalians. So and that's because they are unmyelinated. This is something most researchers never believed in, and I have to fight for 30 years to tell them that this does not look like a guinea pig. This is entirely different. It's unmyelinated. It's glia cells, and this is extremely important because this is probably why we can do cochlear implantation, and that we have such a slow retrograde degeneration when we lose hair cells in humans. And also in the apical part of the human cochlea, we have these units-like assemblies at the low frequency of spinal ganglion, you know, which are surrounded by satellite cells, and they are actually lying together. So this might be very important physiologically, you know, maybe for speech perception, and this has been denied over the years. So much of the experimental work with the spinal ganglion, you know, is not relevant for humans. I think that's important. And this is Fred's temporal bone dissection, you know, where we see the patient, where the patient has lost all the peripheral dendrites, but still the central axons, the central and spinal ganglion cells are still preserved. And how can that be? This cannot happen mostly in animals and in mammals. And it's because of the satellite cells. And we look at the satellite cell, they have something called gap junctions. And these gap junctions, they express connexin 43. So the connexin 43 little molecule is probably the molecule that actually makes these cells preserved, that preserves the cells when you lose the peripheral dendrites. So they can interact and survive. You know, you can have patients with no hair cells, no peripheral dendrite, and you can have a very large population of neurons, which is important for cochlear implantations. So now I will present you to some super resolution microscopy of the human cochlea, and this is a fantastic technique. It's a kind of nanoscopy where you can go down to 18 nanometers resolution, and now you can look at the large molecules. And this needs human cochlea to be embedded and cryosection, which is extremely difficult, as you all know. And this is Dr. Wei Lu who's been in my lab for 11 years. He's a professor and an ear surgeon actually from China, and he is a master of handling this tissue. And here you see a picture of a human cochlear section with the outer hair cell, the inner hair cell stained for paralbumin. And here, the green here is the TMPRSS3, which is actually an enzyme that we know that in mutations and high frequency hearing loss cause non-syndromic recessive deafness and regulate the big potassium channel. And you can see the wonderful resolution, and this is even higher magnification. And now we can see into the stereocilia, we see the enzyme, the protein sitting here in the center, and the actin is red. So this is quite fascinating. And we can now make a very nice analysis of the human organocorti hair cell, the beautiful pillar cells, and the components, the protein contents and structural components, which is very fascinating. And this actually made us to do gene studies to see, can we find genes associated with presbyacusis? And my son, Matthias, who was also in LA, of course, but he was quite small then, he is a geneticist. So he and we perform genome-wide association studies using the UK Biobank, where they have 350,000 people. He identifies 67 loci, and candidate genes from these loci were stained in human cochlear samples. So they were all stained, and we can now, here we can see the result where we can image some of these genes we found, such as LMO7, staining the cuticular plate in the hair cells, LMO7 here in actin, and TRIO-BP in the human stereocilia, which organizes actin filament to rootlets. So they are apparently involved in presbyacusis. And and we also found that these genes implicated processes in the spinal ganglion neuronal cells and involved in neuron plasticity and recovery after trauma. So I think this was published in the BMC last year. So now I want to show you something about the synchrotron images, which is we do together with the Canadian collaborators up in Saskatchewan, and this is a fantastic technique. And you know, with a clinical CT, we have a pretty low resolution. It's a problem. With a micro CT, which you cannot use with patients, you have a higher resolution. You can see a cochlear implant inside here, electrode, and with synchrotron, you have this kind of resolution, much better resolution. And now I will show you a picture I got for last week from Canada. And here you see the last imaging up in Saskatchewan. It actually almost reaches histology. You can see this is how we would like to see the inner ear when we are sitting on the X-ray round with the doctor. We can see the corti tunnel, you know, we see the the organ of corti, different cells, the Reisner's membrane, and so this is a fantastic. This is fantastic, and hopefully it will be used in the future also clinically, so we can see dramatically other things. And here we use a volume rendering software to create 3D reconstructions and tissue segmentation. And here you see we can make sections in at any angles, and we can see into the cochlea with the Reisner's membrane here. We see the cuticle and we see the saccule. And here we see a rendering and staining of the different components of the human inner ear. In this specimen, we see the endolymphatic sac. We see the yellow nerve structures in the meatus, facial nerve. We see the vestibule with the macula, utriculi, sacculi, cochlea, and we even see the folds and middle ear folds. And I will come more back into these folds, chronic otitis media, which is pretty fascinating. And here you see the macula, cuticle macula, and you see macula sacculi also, and facial nerve and carotid. And if you look into meatus, you see, interestingly, with this 3D, you can see that the cranial nerves, when you do the acoustics, we never see that they are really hanged up, these cranial nerves. But with the synchrotron, you can actually see that they are connected with some small trabecles that hangs them up in the arachnoid. Here you see the arachnoid pouch, and here you see the cranial nerves inside them. So this is, you don't see this two-dimensionally on sections, but on 3D you can see. So they are not hanging free, the cranial nerves. We had, I noticed that we had actually one temporal bone that was operated with a wire prosthesis, and we did analysis of this, and you can see how the bone looks, something a little bit eroded here after many years, and we can study the blood vessels that goes into the cruise longest down to the joint, to the stapes joint. So and now this is the most interesting thing, I think, so far. For the first time ever, we were able to see the BAS valve, the utricle endolymphatic valve that connects the pars superior with the pars inferior. And this the red here is actually the BAS valve. It looks very small on histological section, but it is actually quite big. Actually, it's 0.7 millimeter large opening, And if we look deeper into this synchrotron, here you can actually see this for the first time. This 3D, this valve. It looks like a mouse, upside down lip of the mouse, and it seems like a real valve, and that can open under various circumstances. And so I was really surprised to see that it's actually such a big structure. And this has some relevance when we now think about Meniere's disease and the development of endolymphatic hydrops. At the same time, we did, this is actually micro-CT, and we can see the reunion duct, that connects the cochlea with the vestibular organ. It's very thin, very small, which fits with the histology, but usually you see drawings which show quite big channels. So this tells us that an increased pressure in cochlea cannot momentarily lead to an increased pressure in the rest of the labyrinth, because of this very, very thin, less than 0.1 millimeter wide and almost a millimeter long structure. That was very surprising for us. So now we have, I've been working with the SAC for 50 years, and it has clearly a secretory role also, both absorptive and secretory. And there are some papers showing that, you know, of course, Australia, Professor Gibson, who destroyed the sac in patients with severe Meniere's and said that that could have effect. So I was very critical, but maybe there is something to it actually. You know, the Bass valve here, you can see, contains a very soft part, and it's held in place by a small disc here. So when the pressure increases here, which could be the fact when there is a secretion of material in the sac or a reduced resorption in the cochlea, that this one could open up and of course hydrops. This is a possibility that maybe we should think about and think of, and maybe we should consider Gibson's findings and start to do sac ablations in some patients with severe Meniere's disease. So the ear and the endolymphatic duct and sac has a sort of resorptive channel system, which is a little bit like in the eye, you know, where you have the canal de Schlemmey, you have the venous drainage in the angle, and we have a little bit of the same thing also in the endolymphatic sac and duct that can be obstructed and lead to decreased resorption of the sac and lead to endolymphatic hydrops. So when we did the super microscopy and looked at the enzyme distribution, we found, interestingly, that we have evidence that there is both a secretion and an absorption of fluid and sodium in the sac, because we found that there was a polarized expression of sodium-potassium ATPase. Sodium-potassium ATPase, you know, is involved in resorption of sodium and fluid in the kidney, but also in the sac. But they are found also reversely in some of the epithelial cells, which we see in secretory structures, like the choroid plexus. And here you see this structured illumination microscopy. You see the red distribution here of the sodium-potassium ATPase, just like in kidney, in some of the cells, but we also find them apically, suggesting that these cells are secreting endolymph. So the sac seems to have both function. And here you see how they are co-expressed, the molecules sitting in the apical membrane. You see the co-staining, yellow, the green and red. So this is fascinating with this type of microscopy. So more about synchrotron, the synchrotron findings. I guess you all do a round window membrane insertion when you do cochlear implantation and put your electrode. And the base here of the cochlea has a very complicated, complex anatomy. And with a synchrotron and 3D, we can see this very nicely, you know, the different structures. And we can see that the round window is hanged up around, the round window membrane is hanged up around the window with a secondary spiral lamina. And this light blue structure here is the secondary spiral lamina. And that's the one that you often destroy when you do cochleostomy, which is something to consider. The blue here is the, you see the spinal ligament in the cochlea, the lateral wall, and the neural structures. So if we do some virtual cochleostomies, you know, anterior cochleostomy, anterior inferior, we can see that they all destroy parts of this anatomy. The only way is to do it inferior cochleostomy. You can actually come far beneath and do a traumatic insertion of electrode here, but this is also very uncertain. So for hearing preservation, I don't believe cochleostomy is very good. With the synchrotron, we can also look at the vascular anatomy in a different way, 3D. And this is also extremely interesting to see this complicated vascular anatomy of the human cochlea. You know, the arteries inside the cochlea, they come through scala vestibuli, and the veins drain through scala tympani, and separately. And this is very unique, that they actually, the artery and veins go so differently. And this shows the principal supply in the human cochlea based on synchrotron. And then we see that the Rosenthal canal, where the nerves are present, they are supplied differently with vessels, with arteries and veins from the scala tympani and scala vestibuli. So this, I think, explains why you can have such a good implant result in many patients with quite bad anatomy inside the scala in the cochlea, but because you have a preserved spiral ganglion in Rosenthal's canal. I think that's very, very interesting. And here you see the meatus and the blood supply inside the meatus with these inferior vestibular nerve, facial nerve, superior nerve, cochlear nerve. And here we see three-dimensionally for the first time the vestibular cochlear anastomosis, where the neurons pass from the inferior vestibular nerve and sacral nerve into the cochlea. We see these arteries, you know, that supply the inner ear. We have the cochlear vestibular artery, superior vestibular artery, and the cochlear artery. These are the three arteries to the inner ear. Well, now we do the cochlear implantation. What happens with the vestibule? Is it a risk for the vestibular apparatus? Well, yes. Saccule is closest, only some millimeters away from the spiralumina. The reunion duct is very close, which can be damaged. So the round window insertion is not problem-free either. And when we look here now at the synchrotron and the spiral ganglion anatomy, we see an electrode sitting under the basilar membrane nicely. We will see the yellow spiral ganglion, and the spiral ganglion is only 14 millimeter long. The basilar membrane is 34, so it's less than half of the length. And as you know, the number of neurons up here in the second turn, there are in principle 2 turns, is almost 10,000 neurons sitting, you know, in just a 2.5 millimeter range. This is fascinating. And what we did was that we tried to make a frequency localization and an octave band, put the octave bands into this cochlea. And to assess these frequencies, you have, according to the Greenwood formula, so you know you have to know the basilar membrane length. And this is one of the problems to do this in patients from the clinical CT, but I show how it can be done. Here is 10 different human cochlea, and you see the octave bands distribution in these. They are pretty similar, but still there are differences between different persons. The cochlea varies sometimes quite a lot. And here you see the octave bands, the mean octave bands, the basilar membrane, and the mean in the octave bands in the Rosenthal canal. But if you look, usually you use this diameter A as a way to see how big the cochlea is, but it's not a good parameter, because diameter A can be quite small and still you have the longest cochlea. So if you want to assess the different frequencies in your patients to fit the patient the best way, diameter A is not optimal. So the way to do it is if you want to have a patient-specific cochlear implant pitch mapping, which may improve the result, maybe, according to some authors, you can use and assess the number of turns in the human cochlea. So the number of turns is a yellow point here, can be assessed if the radiologists make a 3D reconstruction of the clinical CT. And then you assess the angular distance along the basilar membrane, and you will be able to assess the different frequencies for each electrode and fit the patient. And then you can use this kind of formula, which we published some months ago. But of course, this is a complex way, and you have to automate. So we're trying to get a paradigm and an algorithm for automatic assessment of the different frequencies to make a better implant pitch mapping. Now I will show you also the synchrotron analysis of the folds of the middle ear. This is, as a chronic otitis media surgeon, it's quite interesting to see how the prosthetic space is aerated. Palva's nice work over the years. This gave us a new possibility to look at the aeration pathways. And in these 10 bones, we were lucky enough to see 3 different principal drainage ways or aeration ways in prosthetic space. And here we have reconstructed, or we have segmented the different folds, posterior malleolar folds, the lateral fold, anterior fold, and here is the prosthetic space. Eardrum. Here we can see reconstruct the prosthetic space, how it looks, and this is the other side of the other bone in this person, and we can see here the prosthetic space and how it is aerated in a totally different way, in a medial way. And so actually you can have patients where both ears are aerated quite differently. That could explain why you have patients with a single-side cholesteatoma. I mean, many patients have cholesteatoma only one ear. And here you have, we made a movie and from one of these, and you can see the eardrum is segmented, the tensorfold, the pars flaccida here, the posterior malleolar fold, and we can see how this pouch of von Trölsch is closed, and we can see how the prosthetic space is aerated. And interestingly, there was one of the 9 bones had an anterior aeration. You see a wide aeration, and then nature has created a lamina, a fold back here in the posterior part. So nature doesn't want to have a prosthetic space open on both directions, only one direction. So this is probably very a very nicely aerated space, probably. And you can see that some are quite small openings. You know, you can easily imagine a granulation sitting here in this patient, that you would have a reduced aeration of the prosec space and a retraction of eardrum and the risk for cholesteatoma. Now I thought I will go over a little bit to cell science, to cell anatomy, and tell a little bit our findings of the human cochlear battery, which is the lateral wall and stria vascularis, which has also big implications in cochlear implantations. And we studied the human cochlea. We found this very, very beautiful arrangement of this battery. It's just like a biologic battery with isolators, semiconductors, and pumps. And the green you see here, that is a protein called Claudin 11. It's a barrier that sees that the potassium ions, which are the electric charge carriers, are not going backwards, like they are isolated by this green stuff. And then we have these connexins 26 and 30 you will know about in genetic deafness. They are enormously expressed, and potassium channel, potassium can be transported across the apical turn. And this is a vase confocal image showing the beautiful molecular anatomy of the human lateral cochlear wall. You see here the stria vascularis expressing both, here yellow, sodium potassium ATPase, and this NKCC1, which is also important for potassium shunting. And you can see how variously they are expressed in the ligament, in the fibrocytes, in the lateral wall. It's really fantastic. And now, presbyacusis has now been associated with changes in the fibrocytes expression of sodium potassium ATPase. And here you see nanoscopy. You see the molecules sitting in the cell membranes of the stria vascularis, the red and green and yellow. You can see the different molecules here, the enormous expression of sodium potassium ATPase in the stria vascularis to pump all the time the potassium. And here you see this beautiful Claudin 11, the tight isolator. So when you do cochlear implantation, you easily go into the lateral wall, you destroy this, and you create fistula. And what we see in the humans now is that the connexin molecular structure is not what they say in literature in animals. They are all homomeric, homotypic arrangement like this. This is important considering gene therapy. And now we have look at the genes of the connexin genes, and we can actually spot the genes now with a special technique called RNA scope, which Dr. Liu has developed in my laboratory. And here you see these cells. The red is the cell nuclei. This is the outer sulcus. The outer sulcus is an enormously active area. And you can see the green and yellow here are the connexin 13, connexin 26 molecules. They're all sitting differently, they are not sitting together exactly. They are differently expressed. And here we see for the first time the gap junction B6, which is the connexin 30 gene. Every red spot here you see is a gene, it's a connexin gene. And you can see out the sulcus how enormously they are present in the outer sulcus. So here is a very, very intense absorption of potassium, probably from the organ of Corti after transduction, mechanoelectric transduction. They are shunted through the epithelium up here to the outer sulcus and into the subepithelial space. And if you look in the spinal ganglion, you see this here enormously how these genes are present in the spinal ganglion. So the spinal ganglion, contains this gene, transcribes the genes from the cell nucleus and transport them out to the organ cortex. And you can see that here we have the protein in the membrane in the spinal ganglion. The spinal ganglion cells in human, they can be spike generators because they are unmyelinated. So maybe that's the spike generation here creates the spikes with cochlear replantation. And so here you see a principal drawing showing all these genes present in the spinal ganglion, and you see how selectively they are also expressed in the lateral wall. This is the ATPase, sodium potassium ATPase, which are only expressed in the marginal cells. And the connexin are only expressed in the basal cells and type 1 fibrocytes. This is very nice. Just so time flies so fast, you're tired of me now. Just show a few slides about immune cells. When you put in an electrode, you can activate the immune system, and humans have immune cells. It's not an immune privileged area. And here you have macrophages. You see the green cells here, they're all macrophages sitting, in the cochlea. In the lateral wall, you have a large amount of them. They're very interesting cells. They are very nice cells, because they protect us, but they monitor us, and they can turn to be very evil too. Every neuron, every axon seems to be connected to a macrophage long process. This is fascinating. And here you see the green cells here, and here are the macrophages. And you can see how they penetrate deep into the neurons. They are interacting with the neurons. And here you see this monster macrophage in the spinal ganglion, and with long, small antenna. They are looking, they are searching, they are looking and surveilling, controlling, they are the secret police, looking what's happening. And they can get really angry, and they start the M1 pathway, where they start an inflammation, a destructive inflammation, bad for the inner ear, that destroys cells. But they can also go along the M2 pathway to be saviors and to suppressing T lymphocyte activity, so it's a balance, So when doing a cochlear implantations, one should probably be very careful not to destroy tissue and to start inflammation. And here you see these long processes these macrophages have. They stick it also into the organ or cortex. And if you look with a scanning microscope, you can see the dendritic macrophage is also active hair cells, which seem to be involved in healing after loss of hair cells. Very, very interesting. So a lot of things are going on in there, very subtle differences. And these macrophages, they are hormone dependent. So they are attracted by a hormone or a chemokine called fractalkine, which is the gene which is expressed here in the spinal ganglion. So this is why we have so many of these macrophages in the spinal ganglion probably. Every red dot is a gene for the fractalkine. So we have a complex interaction here between the cochlea and the lymphatic sac, invasion of macrophages, which seem to help us all the time to keep the inner ear clean, and to and if anything is passing into the inner ear, is maybe transported to the sac, where it is destroyed by macrophages that are primed, processed, and information goes back to the cochlea to be effective and prepared for the next time. Oh, now I'm finished, almost finished. I just show the crocodile, one slide. This is a picture. This is a small cubic crocodile. which is the son of Fidel Castro's crocodile, which was donated to Russia and came to Sweden, and the son of it is this crocodile. They had he was so aggressive, you know, they had to terminate it. And now we are looking at the crocodiles to see if we can regenerate hair cells, because crocodile can apparently regenerate hair cells like we know before. But the interesting thing is, you see this beautiful, actually the hearing organ looks like a crocodile, you see? And they have very subtle terminals and very, very nice synaptic terminals with ribbon synapses, very organized, you know, surprisingly. And what we can find is that these probably, they do new hair cell all the time. It's not only when they destroy the hair cells by ototoxic drugs, as they found in chickens and so on, but this seemed to be an ongoing process where we find quite new organelles not shown before, and a gap junction networks that is so extensive, is unbelievable. And they work together with this apparently this new organelle system to make continuous and new hair cells. So maybe we can find out something to use in the humans in the future. I'm sorry to so many slides. I'm sorry. But we say hello from Uppsala, everybody, and from the lab to you all in California. Thank you.
Hello, my name is Dr. Kevin Peng, neurotologist here at the House Institute. Thank you for watching this video. The House Institute provides free educational videos for hearing health professionals worldwide. To help support videos like these and other educational efforts, please consider donating by clicking the link in the description box below. Your generous support allows us to keep videos like these at no cost for you and others. Thank you.